To know that enzymes are important in clinical diagnosis of important metabolites in our body, and to learn the chemistry behind common diagnostic tests, and also to identify normal versus pathological conditions based on these diagnostic tests. This module describes the common clinical tests performed on the human body. These tests are used to obtain physiological parameters that characterize our general health. These parameters can be in terms of the amount of a certain protein, activity of an enzyme, or the levels of a particular metabolite in our body. Each test utilizes a set of specific biochemical reactions to determine the level of a particular parameter. In this module, common biochemical reactions will be described, which are used to diagnose the presence of important proteins, enzymes, and other metabolites in our body. And these biochemical reactions have been established to have direct correlation to our physiological health. These reactions are used as diagnostic tests in clinical laboratories, the outcomes of which are usually in terms of concentration of the parameter being measured. Parameters associated with a healthy condition are usually expressed as a range of concentration. An individual is diagnosed as healthy or pathological with respect to a parameter depending upon whether the concentration of the measured parameter falls within the normal range or lies above or below the normal range. Along with the relevant diagnostic tests, we also describe the possible pathology associated with the abnormal parameter levels. We'll first talk about enzymes used as reagents. Certain metabolites in our body are important indicators of our physiological health. Estimation of the concentration of these metabolites is achieved by enzymatic reactions in the clinical lab. Reactions that we'll be talking about describe the estimation of metabolites and proteins important for the function of key organs like liver and kidney. Urine and blood are usually the source of the samples used in these estimations. All the tests from urine and serum, samples from the liver and kidney are collectively called as liver function tests and kidney function tests. We'll talk about the estimation of urea from serum using urease and glutamate dehydrogenase enzymes. Urea is an important parameter that is checked for healthy kidney function. Estimation of urea acid in the serum uses an enzymatic reaction which yields a colored substance that is spectrophotometrically quantified. The enzyme urease catalyzes the reactions shown on your screen. Urea is subjected to the action of urease enzyme under acidic conditions. This yields ammonium ion, which is further reacted with alpha ketoglutarate in the presence of NADH and the enzyme glutamate dehydrogenase. NADH is oxidized in this reaction, yielding NAD+. The reaction is monitored by spectrophotometric quantification of NADH at 340 nanometers. Normal levels of urea in adults is 6 to 21 milligram per dl in females and 8 to 24 milligrams per dl in males. Now we'll talk about estimation of uric acid from serum using uricase enzyme and peroxidase enzyme. Uric acid is a product of purine metabolism, that is, of adenine and guanine in the body, that is filtered by our kidneys. Higher than normal levels of uric acid in the urine is indicative of kidney damage. The enzymatic reactions shown on your screen are used to estimate the levels of uric acid. The uricase enzyme acts in the uric acid in the serum sample to give allantoin and hydrogen peroxide. The hydrogen peroxide then acts with amino antipyrin in the presence of peroxidase enzyme to give a red dye called as quinone imine. The red dye produced in the above reaction is colorimetrically estimated at 520 nanometers and correlated with the levels of uric acid. We'll now talk about the enzymes used in immunological assays. The specificity of enzymatic reactions makes them useful as reagents in immunological assays that require precise detection of substances in a clinical sample. Immunological assays involve detecting the presence of an antigen or an antibody in clinical samples. Usually, sensitivity of detection is a major concern in such assays. It is desirable to have systems which can detect low levels of antigen or antibody and at the same time give a signal output that is well within the detection range of the instrument being used. ELISA or enzyme-linked immunosorbent assay is a typical immunological assay. In this assay, an antigen or an antibody is detected in a clinical sample. An antigen is immobilized onto a solid support such as an ELISA microtiter plate. A clinical sample is then added onto the plate and allowed to incubate so that the antibodies in the clinical sample that are specific to the coated antigen on the plate bind to the coated antigen. Successive washings remove the unbound and non-specific antibodies from the plate, leaving behind only the coated antigen bound to the specific antibody. This antibody is also called as a primary antibody in the clinical sample. The next step is to now add a secondary antibody to bind to this primary antibody. Secondary antibody of this kind is usually conjugated to a specific enzyme. A carefully chosen substrate to this enzyme is then added to the ELISA plate to allow the enzyme action to take place. The substrate is carefully chosen such that the products of the enzyme action is a colored compound to allow colorimetric estimation of the enzyme action. In this system, the activity of the enzyme is reflected in the intensity of the colored product. This can be correlated to the amount of primary antibody. A commonly used enzyme substrate system is the horseradish peroxidase and diaminobenzidine system. A brown colored product is obtained. Other enzyme substrate combinations include substrates that are cleaved to give a fluorescent product. The fluorescence of the fluorescent substance will be much more than the intensity of the colored compound in the previously mentioned HRP-DAB system for the same level of antibody in the clinical sample, and thus the output signal will be much more amplified. This makes detection much easier to measure and, more importantly, allows very dilute amounts of antibody to be easily detected in the sample. This is an example wherein enzymes can serve to amplify the signal in an important clinical assay, improving the sensitivity of the assay. Some important reactions of clinical significance are such that they estimate the levels of important enzymes. Levels of certain enzymes in key organs such as the kidney and liver serve as indicators of good physiological health. The following part of the module will discuss what are these enzymes, how they are estimated, and what are their normal levels in our body. We'll first talk about the estimation of alkaline and acid phosphatase enzymes. Phosphatases in the cell act on the organic phosphate esters releasing phosphate. They are classified into two categories based on their pH optima: the alkaline phosphatase and the acid phosphatase. Alkaline phosphatase helps in transporting phosphate groups across cell membranes. Commonly used substrates for this enzyme in our blood is paranitrophenol phosphate, abbreviated usually as PNPP. Hydrolysis of this substrate yields paranitrophenol, which is ionized at alkaline pH to a yellow colored substance. Paranitrophenol is estimated colorimetrically at 405 nanometers and its concentration is correlated with that of alkaline phosphatase in the blood sample. The enzyme activity is usually expressed in international units, that is, micromoles of paranitrophenol released per milliliter per minute. Acid phosphatase hydrolyzes aliphatic, aromatic or heterocyclic phosphoric acid esters between pH 4 and pH 6. This enzyme is secreted by white blood cells, red blood cells, platelets and prostrate cells. The enzymatic estimation for both acid phosphatase and alkaline phosphatase uses the same substrate para-nitrophenol phosphate or PNPP. In case of acid phosphatase, the reaction is carried out between pH 4 and 6 and hence para-nitrophenol released is colorless as we have an acidic condition. Addition of sodium carbonate bicarbonate buffer of pH 10 converts it to a nitrophenolate ion, which is a yellow colored compound. This can then be acid colorimetrically and correlated with the acid phosphatase enzymatic activity. The normal range of alkaline phosphatase in the blood is 20 to 140 international units per liter for adults. Higher values are found in children and pregnant women. Besides other causes, abnormal levels of alkaline phosphatase in the blood broadly indicates either damaged liver cells or defects in bone growth. Now we'll talk about the estimation of two very important enzymes, the SGOT and the SGPT enzymes. SGOT enzyme, also called as serum glutamate oxaloacetate transaminase, and the SGPT enzyme is commonly called as the glutamate pyruvate transaminase enzyme. These two enzymes are released in our bloodstream upon liver damage. Increased levels of these enzymes are thus indicative of liver damage. The reaction catalyzed by SGOT is, as shown on the screen, aspartate and alpha ketoglutarate react to give oxaloacetate and glutamate. In the clinical estimation of SGOT, the oxaloacetate released in this reaction is estimated by reacting it with 2,4-dinitrophenylhydrazine. The end product of this reaction is a brown colored product which can be estimated colorimetrically at 505 nanometers and correlated to the activity of SGOT. The reaction catalyzed by SGPT is alanine plus alpha ketoglutarate giving rise to pyruvate and glutamate. In the clinical estimation of SGPT enzyme, the pyruvate released in this particular reaction is estimated by reacting it with 2,4-dinitrophenylhydrazine. The end product of this reaction is a pink colored product. The pink colored product obtained as a result of this reaction is colorimetrically estimated at 505 nanometers and correlated to the activity of SGPT. The normal range for the levels of these two enzymes in the blood is, for SGPT, it is 10 to 40 international units per liter in adults and 7 to 40 international units per liter in children. For SGOT, it is 8 to 20 international units per liter in adults and 9 to 8 international units per liter in infants. These values are subject to variation with respect to age, gender, and body weight. Levels beyond this can be indicative of damage caused to the liver cells either due to cirrhosis, hepatitis, or toxic liver necrosis or cancer. Now we'll talk about how to estimate another enzyme in the serum, which is the lactate dehydrogenase enzyme. Lactate dehydrogenase enzyme, or LDH, catalyzes a reaction in our body which is pyruvate combining with NADH in the presence of hydrogen ions to give lactate and NAD+. It is present in large amounts in blood cells and heart muscle. It converts pyruvate, which is the final product of glycolysis, to lactate in the absence of oxygen. When lactate is in high concentration, it exhibits feedback inhibition. When cells undergo injury or damage, LDH is released from the cells into the serum. Levels of LDH in the serum rise to higher than normal levels during certain disease conditions such as myocardial infarction, hemolytic anemia, pernicious anemia, or certain infectious diseases such as meningitis, HIV. Lactate dehydrogenase levels are used as marker enzymes to monitor such conditions. Now we'll talk about how to estimate cholesterol. Enzymatic estimation of cholesterol from the blood serum is based on the hydrolysis of the cholesterol ester found in the blood. Cholesterol esterase is used to hydrolyze the esterified cholesterol into cholesterol and fatty acids. Cholesterol oxidase acts on the total cholesterol to form cholest 4N3-one and hydrogen peroxide. The released hydrogen peroxide is degraded by the peroxidase enzyme or POD into water and molecular oxygen. A substrate 4-aminotyrene then reacts with the molecular oxygen to give a red product quinone amine, which is a dye that can be colorimetrically estimated at 560 nanometers. Normal value of serum cholesterol is 100 to 200 mg%. Higher cholesterol levels can indicate risk of coronary heart disease, while lower than normal levels are indicative of anemic conditions. Now we'll talk about how to estimate glucose in the blood. Blood glucose is measured by the glucose oxidase test. In this test, the blood sample is reacted with glucose oxidase enzyme, which converts the glucose to gluconic acid and hydrogen peroxide. The hydrogen peroxide is reacted with peroxidase enzyme in presence of a 4-O-dianicidine. This yields a pink color that can be measured spectrophotometrically at 540 nanometers. The intensity of the pink color is correlated with the levels of glucose in the blood. Acceptable levels of fasting glucose in the blood is 80 to 100 mg%. Fasting glucose levels exceeding 100 mg% are indicators of hypoglycemia. In contrast, blood glucose levels lower than 80 mg% indicate hypoglycemia. The figure on your screen shows you the various reactions that are involved in the glucose estimation test. Now we'll summarize the various points covered in this module. First of all, enzymes are important in clinical diagnosis of important metabolites in our body and can be used as reagents as well. Enzymes which are of clinical significance are estimated, and enzymatic estimation of cholesterol from the blood serum is a common test. Enzymatic estimation of blood glucose is measured by the famous glucose oxidase test.